The brain requires a lot of genes to get put together. We have about 20,000 genes in total in our genome, and the brain uses almost half of them. And so that creates a lot of potential problems, because if almost any one of those genes is not working properly, we can develop disorders of the brain like intellectual problems or cognitive problems.
Our intellectual cognitive abilities, language, writing, speech, is much different than our non-human primate ancestors. And that's due to changes in the DNA that then impact the function and complexity of the brain. But along with that are changes in DNA that causes diseases.
There are many types of mutations, and we have a long way to understand the effect of most of them. We've had better success in identifying the mutations that impair genes, because that's obvious. You've got someone who suffers from a disease and you study their DNA and you found a mutation, you can relate that to the cause. But we have a long way to identify as many mutations as possible that actually might confer protection and might improve health.
Function of the ataxin-1 gene, its primary function is to encode and give the genetic information for the ataxin-1 protein. And when we talk about function of the gene, what we're really talking about in genes like ataxin-1 is we're talking about the function of the protein. And that's one of the major questions that we have tried to address over the years, is because we think the function, the normal function, if you will, of the ataxin-1 protein is very much linked to the molecular aspects of why the mutation in that protein then causes the disease, spinocerebellar ataxia type 1. And what we know so far is that an important function of the ataxin-1 protein is to regulate gene expression in a variety of cell types, not just the brain, but also some of the muscle and other tissues in the body. And what we're trying to understand is why certain neurons are more susceptible to the mutation that disrupts that function or alters that function than other cells. And that's an interesting biological question, but also perhaps in the root of answering that question might give us better information of how to counteract the negative aspects of the disease-causing mutation in the gene/protein. But at this time, we're focusing on the nucleus of the cells and regulation of gene expression in terms of its function.
In 2003, the Human Genome Project determined that each human has 6 billion letters in its DNA, 3 billion base pairs inherited from their parents.
Each time we conceive a child, out of the 6 billion letters, there are actually about 60 that will change from parents to the child. Most of them have no effect. Some may have very mild effects, good or bad, and rarely, but not so rarely, it can lead to a dysfunction in a gene that is very important for the brain function. Many of them are not transmitted. Some of them are transmitted. Fragile X can be transmitted in a pretty bizarre way. But because there are new mutations happening at each generation, each time we conceive a new human, then it will stay this background of people with such mutation. And this is something we cannot really prevent unless the only way would be to sequence in every fetus, and then not keep those who have mutations that we can identify as very bad. But this would pose both ethical questions, because we don't always understand what a mutation effect is. And of course, for the moment, it's not feasible or desirable.
I think one interesting area that we're aiming for is to try to understand how the development of our brain predisposes us to various disorders that might develop much later in life. So at the moment, we know enough to know that the development of the brain can predispose to certain conditions that affect a child, like focal epilepsy or intellectual disability or autism spectrum disorders. We also know that there are roles for genes in creating other disorders that present late in life, like degenerative conditions or Alzheimer's disease. What we don't yet know, and I think we might understand more about soon, is how the pattern of development also contributes to what happens later in life. Not just the genes, but in fact these exposures and so forth that can also alter our genome in a way that happens independent of what we inherit.
I work on childhood neurodevelopmental disorders. In particular, one such disorder is called Rett syndrome. And that disease shows up much earlier in life. It typically shows up in the second year of life, where the babies lose the skills they used to do and cannot speak and cannot learn new things and will not use their hands anymore. They will wring their hands and they have balance problem, but it's because of changes in the function of the brain early on. That disease is very different from the ataxia because it's only one in a family. The ataxia, I told you, it passed from generation to generation, so you have many affected individuals. In the case of Rett syndrome, there'll be a couple who will have a child who's healthy, and then the next child may have Rett syndrome. Or maybe another couple, their first child will have Rett syndrome, but then they have other healthy children. So it's just one in a family, but it's sporadic. It's not inherited. Parents are healthy, child is affected, because in the sperm, in one sperm, out of the many, many healthy sperms that that father carries, one sperm will have a mutation. And if that sperm gets fertilized and becomes an embryo and then a child, that will bring on Rett syndrome. So this is a class of disorders which are genetic, but not inherited. So it's still caused by a gene, but it's a random mutation that happens.
Please welcome on stage Huda Zoghbi, Harry Orr, Jean-Louis Mandel, and Christopher Walsh.